So welcome to our today's seminar. Our speaker is Dr. Julie Peterson from University of Nebraska-Lincoln. Dr. Peterson supposed to be here this spring, but I don't believe I'm saying that due to the Florida snowfall, he can make it. But we are glad that she is here. Dr. Peterson got her bachelor's from Ohio Wesleyan University in zoology, and then she got her PhD from University of Kentucky and got her postdoctoral experience from University of Minnesota. She joined University of Nebraska in 2012 as a faculty member, and now she is a full professor and extension specialist, and also the interim director of her research, extension, and education center. We are really glad that she is here today and she has some exciting career update which she will share with us. Before starting the seminar, I have a quick announcement. Dr. Dan Hunt will be taking Dr. Peterson to dinner, and faculty members are welcome to join, and please contact Dr. Hunt if you are interested. And secondly, we'd like to thank Dr. Norm Lepla and his wife Carol Lepla for helping us in Dr. Peterson's lodging situation, because due to the green weekend, there is nothing available around here. So thank you so much, Dr. Lepla and Carol.
You will get an invoice.
I don't know about that. Game day rate?
Great.
Good response to service. Can everybody see and hear online? Is that good for you guys? Okay. Well, yeah, right. I grew up in a state that loves their college basketball. I live now in a state that loves their college football. The stadium in Lincoln for UNL football is the third largest city in Nebraska on game days. So even with that context, I'm super impressed with UF fans and the lack of hotel rooms for 100-mile radius around. But thank you guys so much. Thank you to Tafsir, to the students for inviting me. I honestly feel like that's like the most impactful thing, like no offense faculty members, but to be invited by the student group is really, really meaningful to me. I appreciate that very much. Also, as mentioned, I really appreciate everyone that has helped me on this second attempt to get to Gainesville, because yes, in January, I thought this was such a clever idea. I had a meeting already in Pensacola Beach in January, and I was going to rent a car and drive from Pensacola Beach to Gainesville, which should not be a problem, right? But no, we had that record snowfall in Pensacola Beach, and the bridges were closed, and we were literally stuck on the island during this NC 246, the USDA multi-state group for corn management. So here we are really enjoying that weather, and I did have my family with me too, and so this was not built by my kids, but my kids did enjoy going out and playing in the snow and on the beach at the same time, which was a little bit of a crazy experience. So thank you everybody for helping me get back here. So yeah, I thank you so much for the introduction. I am from Kentucky. I grew up in Louisville, Kentucky. Went up to Ohio for my undergrad and then came back for my PhD at University of Kentucky. Got to work with genetically modified crops. It was a little bit in the early days of BT, of Bacillus thuringiensis. And we're still asking a lot of questions about risk assessment, right? We put out these proteins into the environment that are insecticidal proteins. They are quite specific. But can there be unintended consequences if these insecticidal proteins are moving through the food web? So I got to work with ground beetles and spiders and kind of track movement of BT proteins through the food web, and do some cool work with spiders. I've already been hearing, I know there's some spider fans in Florida, and got to work with these really cool linnaeid spiders and look at them eating corn pollen, which was not something that was documented, but we actually showed that they would like it would get caught in their webs, they'd seek out the pollen and eat it, and it did have a survival benefit for these linyphiid spiders as a supplement to prey. I then did go up north to University of Minnesota. I barely survived 2 winters up there. It was a bit much, but loved my time there. I got to work with Dr. George Heinkel, and I worked with conservation biological control for soybean aphid. And I worked with some unique systems with some biofuel. These are fast growing willows that were being cut and used for cellulosic ethanol production. And then like mixed prairies that were also intended to be harvested and used for biofuel production. So just kind of looking at, can this habitat modification assist with soybean aphid biological control in adjacent soybeans, looking a lot at the Harmonia lady beetle, working with some of the parasitoids for soybean aphid. And working a lot with a USDA SARE project for that. So yeah, then in 2012, I came to, 2012 I started at Minnesota. 2014, I came to University of Nebraska-Lincoln. For most of my time there, I've been in a 50-50 extension and research position. And so you'll kind of see that in my talk today. Like, it's not really a heavy research presentation. I'm going to talk a lot about extension and kind of approaches there too. And I am based about 3.5 hours west of the main campus. So I am at our West Central Research Extension and Education Center out here in North Platte. And we're quite arid. We get about 18 inches of rain a year. So the corn production and a lot of the row crop production is irrigated. We have center pivot irrigation. So you see from the airplane all of the circles, that's what that is. We do experience, pretty cold winters and a lot of wind, but, do have kind of the 4 seasons out there. And then, yes, I have been the interim director at that center since June. So that was like a little bit of a surprise administrative position I was asked to take on, and I've been trying to balance that with my program for the last couple months. And then my very last slide, that'll be the teaser. I do have totally different career update that I'll share on my very last slide. So the title that I kind of shared is this people and ecosystems focused approach. And just to kind of share a little bit about what that means to me of over the last 12 to 14 years is focusing on pest management through the ecosystem lens, that we're managing economic pests, but we're also doing a lot to conserve beneficial arthropods and promoting ecosystem services more broadly. I really appreciate collaborating with folks who are also looking at like pollination services, biofuels, if we can also maybe graze livestock, trying to look at it as a systems level, and reducing insecticide use through increasing efficacy and efficiency, but also decreasing non-target effects. I also try to approach this through a people-focused lens as well. And that does really come a lot from having this, at least half of my appointment has been extension. And so really having stakeholder needs driving the research, communicating results back to impact agricultural practices, and really integrating students and scholars in my program with the extension side of things as well. And just trying to, provide a welcoming and supportive and balanced environment in our program for the people who are training and coming through. So what that's looked like, we call my lab the Agro-Ecosystems Entomology Lab. Things always start with needs assessments, observations, and concerns from stakeholders. So we've had, research objectives that came from some crazy questions someone asked me after an extension talk. And I'm like, I don't know how to answer that question. Like, oh, maybe that should be an undergrad project next summer. Like, let's look into that. Those lead to the research objectives. I do, try to pull from the kind of traditional pillars of IPM. Maybe this might be a little sacrilege for corn IPM. I'm putting transgenic as kind of our host plant resistance pillar here. And I'm not going to read all of these things. It just kind of this kind of just shows, bulleted a few of the different types of projects that we have done within these different pillars, but also, of course, that these things really need to be working together in synergy and collaboration with each other. And then the end is always going to be these results being communicated back out with the scientific peer community, but especially that extension stakeholder. And another way to look at this, I kind of think of extension as the bookends of an applied IPM program. And that's sort of, again, this needs assessment, starting everything out, and then the communication back with stakeholders being at the end point. And then I won't go through all of these, but just sort of like a bit of the library of IPM, right? We're doing a lot of monitoring and ID, decision tools, economic thresholds, and then our sort of our pillars of IPM as well. And I'll kind of talk a little bit about some different projects that touch on these different things. For today, I do want to focus on one particular insect with you guys. So this is the Western bean cutworm, Stryocosta albicosta. It is a noctuid. It is a pest of corn and dry edible beans. So this is actually really off the presses enough that I didn't kind of work it into the presentation today, but that, right, you think about a pest that is on corn and dry edible beans. We did work together with collaborators in Mexico and found that this is a historically out of Mesoamerica species, that it moved up with the three sisters, corn, beans, and squash system, likely with early indigenous groups that moved up into the southwestern United States out of Mesoamerica. And that's been super fascinating. This species was thought to be more native to the western and central parts of the US, North America, but it really actually did originate more out of southern Mexico, Mesoamerica. So that was sort of an aside of just like, I always find it really interesting when pests are, say like Dibrotica beetles that are on corn and squashes, and then this is, one on corn and beans. It does feed on the ears of the developing corn, can cause extensive feeding damage, not just feeding on the kernels, but also allowing in secondary fungal infections into the ear. We can get these ear rots, and these can cause mycotoxins that are really dangerous, especially if, like many people in Nebraska in the central US, are trying to feed this corn to cattle or other livestock. That can be very dangerous. One thing that's different, I know I've talked to at least one person who works with Spodoptera through Japerta, but if you work with other caterpillars, you probably know you can't rear them together. They will eat each other. Especially if it's like a Helicoverpazea, corn earworm, they're so vicious, right? These are really wimpy. They're not fighters. Western bean cutworm is not often cannibalistic. It can be if you put it in terrible situations. But this is a real ear that really had 4 mature larvae feeding on it. And they were all kind of at the tip of the ear together, just feeding and being just fine with each other. But they will get absolutely decimated by corn earworm and fall armyworm. They're not competitive against these other species. It is estimated as the #2 yield loss causer of corn in Nebraska and also in Ontario, Canada. And I will tell you about that right now. So I mentioned sort of the belief for a long time has been that this gray area was the ancestral historic range of Western bean cutworm. We can now say that this has been the modern range of Western bean cutworm. And the economic damage was really limited to just sort of the Western Corn Belt. And so I am like here in the center of the epicenter of this area. However, oh, there I am. It has spread through Eastern Corn Belt since 1999. That may, if you ever have heard of Western bean cutworm, this might be why. Around that time, it did this huge spread eastward, and there was, of course, a lot of focus on it. Like, what is this? Where is it coming from? Why is it spreading so suddenly? It since then has sort of settled in to a few patchy areas. It has not established in this whole area, but it has settled in areas like southern Ontario and all along the Great Lakes. So kind of the counties of like Indiana, Ohio, Michigan that touch the Great Lakes have like established populations now. And why is that? So one of the really interesting things with this expansion, this hypothesis that came out, was like, okay, what happened around leading up to 1999, 2000? The introduction of BT corn was in 1996. The first BT corn that came out expressed this Cry1AB protein. This protein will kill corn earworm and other ear-feeding caterpillars, but it has no effect against Western cutworm. So essentially the thought is that, I'm going ahead on this. Essentially the thought is that by removing these competitors, these very aggressive corn earworms that would eat a Western bean cutworm normally, by removing them from that ear-feeding niche, it opened up this niche for Western bean cutworm to be able to move eastward across more of the main corn belt. So we do have some different genetic evaluations that say like this eastward expansion did all come from the Western Great Plains. And then we've done some modeling with MaxInt as well to try to get a better idea of what are the commonalities of the geographies where it has established. And there's a variety of different things. Kind of you need to have cold winters and warm summers. You need to have some dry periods. You need to have colder nights and warmer days. But a big thing was sand content of soil. And that might not be surprising if you think about those counties that are along the Great Lakes are going to have much sandier soils. And actually, Western Nebraska has super sandy soils too. So I'm going to talk about its life cycle now, and you'll see why sandy soil matters. So in the wintertime, the Western bean cutworm will actually dig down into the soil about 4 to 6 inches deep and make a little chamber. And it spends the entire winter and into the spring as a pre-pupa. Making its chamber is very important. It uses its saliva and kind of makes like a little concrete wall around itself. And that can protect it from entomopathogens and temperature fluxes and even protect it from drowning if we have a wet spring. And they can go deeper in the soil and make a better chamber in sandier soil. So really it's that overwintering survival is much better in sandy soil. In June, they will pupate and they will emerge as a moth. They will mate and fly into corn or dry beans. She lays her eggs all together in an egg mass. And then the young larvae will hatch out. They climb up vertically on the plant, and they actually feed up on the tassel tissue, the male reproductive structures at the very top of the plant, for several instars before they move back down. And there's some interesting hypotheses around that. The tassel tissue has more protein compared to the ear of corn, and we think that's actually related to that cannibalism. Right, so corn earworm and other caterpillars can get a great dose of protein by killing and eating other caterpillars. But since this species doesn't do that, it is getting its protein to complete its life cycle from that tassel tissue. They then move down into the ear. They feed on pollen kind of as they're moving down, and then they move into the ear and complete that larval development. on the ear, and then they drop off and do that burrowing down into the soil. So what are the different ways we can manage this? I mentioned already not all BT proteins work. The ones that ever worked were only two of them, Cry1F and VIP3A. And we also then do, like, so that would be sort of the, you know, at the time of planting, that could be a decision of choosing your BT traits. As the moths start to fly, we do use degree day models to predict flight. And we do pheromone trap and black light trap monitoring. This is a pest that needs to be heavily scouted. We recommend a lot of scouting and use of an economic threshold. If between 5 to 8% of plants in a field are infested with eggs or larvae, and it has no BT protection, we would recommend foliar applications of insecticides. And there is a window if these applications are not made before the caterpillars enter the ear. Of course, they're protected inside of the husks. So we have done quite a bit of work. When I first started at Nebraska, I was hearing from a lot of people, this protein's not working anymore. And so that was our first big project as we were looking into the Cry1F protein. And we can very safely say now there's a resistance evolved to the Cry1F protein. That has been removed from all the labels for this insect. And we really only have the VIP3A protein remaining. But we're even concerned about that one. There's work we're doing that I don't have time to talk about today, but that protein is expressed in a blended refuge scenario. So it's like the seeds are a mixture of BT and non-BT. And if you think about it, the refuge plant, that female plant, is actually being pollinated by BT plants all around it. So the ear is not a true non-BT refuge ear. That ear is expressing a mixture of BT and non-BT, and even each kernel can be like this hybrid of expression and low levels of expression that may actually make resistance happen faster. So that is something we're concerned about. We kind of have one man standing in our defense, and it's a little shaky. So, you know, that is something I have really tried to work on a lot, is trying to look at what are other management options we can present here. So we have done quite a bit of work with some trichogramma species for egg parasitism, as well as looking at generalist predators that would feed on the eggs and larvae, such as lady beetles. And we also have done a lot of work with the foliar insecticides as well. We also have had a lot of concerns about timing, about resistance, and have looked into a lot of different things with like application methods. How can we really maximize efficacy and efficiency here? And what I'm going to focus on a lot today is going to be like that connection between the natural enemies and less toxic insecticide use. So kind of when I'm just kind of approach this from a bit of like, what's the process, this people and ecosystems, this extension driven, and that step one is needs assessment. So fairly early on in my time in Nebraska, we did kind of start forming these connections with key audiences, and we did engage in more formal, this is what I wanted, we did engage in some more formalized survey tools to determine knowledge gaps and set research priorities. There it is. Sorry, this was the slide I was looking for, the actual results of that survey. So we did this survey with my former master's student, Weston, and we were really trying to ask, what are the stakeholders' main concerns regarding Western bean cutworm management? What are their current practices? Where are gaps in knowledge? This survey was delivered to crop consultants and corn growers. We do have a very robust crop consultant community in the row crop industry in Nebraska. So these are agricultural professionals. Many of them have degrees in agronomy, and they're being hired by the farmers to conduct scouting of their fields, to give them advice. if they need to spray, what should they spray when. So these are very, specialized focused people who are really experts in this area. We got pretty good responses for our survey, and we were asking them about their experiences with farming between 2014 and 2016. And we're happy to get that published in Journal of Integrated Pest Management. But the critical results here was kind of echoing these concerns with Bt traits. Most crop consultants were saying this Cry1F protein is not working like it used to. The BIP3A are working, but at that time they were very limited in commercial availability. I'll say that is one thing that has changed since the timing of this survey. There were a lot of concerns with chemical control as well. Again, kind of just over the majority of crop consultants felt like insecticides weren't working as well as they used to against this insect. And they were using pyrethroids, that class of insecticides, very, very often. So 80% of the insecticides targeting this insect were from this single class. And there was really very little rotation in the modes of action being used. However, our results really also showed this hope and opportunity for integrating biological control into this system. So about 10% of people said, I am using insecticides that are less toxic to predators already in my operations. But here's where I saw that ability to move the needle. About a third of them said they would use less toxic insecticides if they knew which ones they were. And so this kind of gave us two things to go on. Not only did we already know a little bit about which ones would be less toxic, and so like that was just an educational gap. So like, all right, we already know some information to answer this. We just need to share it with you better. But we also said, well, there's a lot more we can do in this area. We can study this a lot more and give better recommendations for what products would be less toxic to natural enemies. And that's an area we really wanted to pursue based on these survey results. So that really did kind of get us thinking about biological control for Western bean cutworm. There was some existing literature, but it was sort of, getting natural enemies and putting them in a petri dish in the lab, a little bit older literature. But we still had the question, well, what is present at the right time out in the field eating Western bean cutworm? Then what can we do to support them? And the two things I'm going to talk about today is improving habitat and reducing non-target effects of insecticides. So we do have a pretty robust ability to see these different field observations of if you're out scouting, you're going to come across some of your typical generalist predators. You're going to come across your lady beetles, your aureus, both the adults and a nymph there. I like this as like family dinner between the adult and the nymph. You're also going to come across green lacewing larvae. I love this photo because if you can see some of these eggs, it's like a beach ball that's been deflated, right? The chrysopid, the green lacewing larva, has pierced that egg with its cool sickle mandibles and has sucked out the juices of that egg, and it's like little shrunken beach ball now. We also did some fun things of, like we are obviously not seeing all of the things happening in the field. So we put out some time-lapse cameras with our intern, Gloria. These are really cool time-lapse cameras that we put out, trained them onto an egg mass in the field and looked at these recordings and it saw predation events happen. And I like this one because this is an aureus that came back at the same time 2 days in a row. These are 2 different days, but really similar time of day. So that was really interesting. There's some foraging behavioral patterns of this aureus here. And then at the end, all these eggs were disturbed and or fed on, and it really reduced the egg hatch ability of that egg mass. And it came back twice in 2 days and it stayed there for 2 to 3 hours at a time. It was really feeding quite a lot. So we also have done some field surveys for natural enemies, just sort of your typical kind of abundance collections, and really just identified that our key ones in these cornfields were Pipidamia convergens, Poliomygilla maculata, green lacewings, and aureus. We then also did some simple molecular gut content analysis. So we collect predators from the field, we extract their DNA, we design PCR primers that are specific to the Western bean cutworm, that pest species. And it is just a simple yes, no, kind of the most basic PCR you can do. And get yes, no's, right? Did that predator have Western bean cutworm DNA in its guts at time of collection or not? Did it have detectable DNA at that time? Because there are, of course, some different things you do to determine what is your detection window using this PCR tool, that kind of thing. So that really, what I just want to point out is that that kind of highlighted some different things of, maybe not surprisingly, there were some very common predators that kind of overall contributed the most number of positives, yet the percentage within that species was lower. And then there were some less common predators, like our nabids or damsel bugs, that were not as common in the fields, yet they provided a higher percentage within that species. So there sort of presents kind of two different approaches of what can we do to support the most common one that is probably functionally having the biggest impact right now. And also, what can we think about in the future? Are there ways to enhance that navid population if they as a species prefer this pest more? So a couple of kind of interesting approaches came out from that. This is kind of now stepping to the habitat management. So now we know a little bit more about what are the key natural enemies. And now we want to think about how do we actually enhance and support them. So one of the projects that I worked with is with a program called Corners for Wildlife. I talked about how in my part of Nebraska, we have the center pivot irrigation. So that's like 132 acres, that's something like 56 hectares of irrigated crop. And that's going to be probably corn on corn on corn. But then there's the corners of that square that circle of irrigation doesn't reach. And the question is, what do you do with the corners? And some farmers are like, more corn in the corners. but I'm excited to work with some conservation partners to encourage them to plant perennial native habitat that also can attract pheasants, because Nebraskans love to hunt pheasants, and that's a great umbrella conservation. And so we worked with these like the Nebraska Game and Parks, Pheasants Forever, Nebraska Environmental Trust, and we did some surveying. So it's a unique conservation program. We did some surveying of could these perennial habitats next to your cornfields help you to support lady beetles and other natural enemies that could then eat Western bean cutworm in your cornfields. I think I was overambitious about the number of slides I included for this project. I will tell you that we did a bunch of different things and looked at not just like lady beetles, but what I'll show you for right now is going to be the lady beetles. This is what that design looks like. You can kind of see the aerial view of what the center pivots and the corners look like. And I think I will just show you kind of the key for this point is we put out these sentinel egg masks, right? So these are like just, it's hard to come across. It's hard We have a better system now, but it's hard to like produce Western bean covering egg masses and use them for this in the field. So in this, we use the Festia cuniella as a proxy for that and put these egg cards out in the field. And you count the number of eggs before and after. You have a control to account for if there was anything washed off by rain or irrigation or wind. And you compare what was that reduction in the number of eggs during the time they were out in the field. So that is what I would like to show is comparing high diversity corn edge. This is the corn that is next to those conservation perennial habitats compared to, say, any of the other locations, which is corn like very, very far inside, and then corn on the edge very far inside when it just had more corn at the corner. That's LD, low diversity corner. And not at both dates, but at least on the first date that we put out these sentinel egg masses, we did have this one significant effect of next to the perennial conservation habitat, we saw a higher percent reduction in those sentinel eggs. And we do think that that really was based on lady beetle predation. So we did also incorporate the predation type, right? You can look at the eggs under the microscope. If it has a ragged corian, that means it was fed on by a chewing mouthpart predator. And that was the majority, especially during the earlier time when we had the significant result. We did also see that deflated corian, like I brought up before, some of them had that deflated beach ball look that was due to piercing, sucking mouth parts. So in this, we really did feel like the lady beetles were driving it. And this was one that this is one of our study sentinel egg masses literally caught in the act as we went to collect it from the field. It was on it. And then I got my phone out real quick and tried to take a picture of it. So sorry, I think I will skip through this, but just to say, we also saw some benefits for dung beetles and pollinators and birds. And the pheasant conservation in this case was a nice umbrella conservation for arthropods providing ecosystem services. And we've continued this work with some really cool things, working with some wildlife biologists, kind of looking at this, but on a little bit bigger scale of larger acreages of CRP, that's like the Conservation Enrollment Program, next to crop fields. Okay, so the other aspect, right? I said, like, what can we do for the habitat for natural enemies? What can we do about insecticide use? Can we look for active ingredients and insecticides that have fewer impacts on the natural enemies? We now know a little bit more about who are the key players, who are the key natural enemies. And this is something we've done in a variety of ways. We do work with industry a lot to do different efficacy spray trials. We always add in natural enemy counts to those types of studies. We've also done some different toxicology studies, what are kind of digging a little further into what's the lethal and sublethal impacts. Doing some economic comparisons, because this is something I was chatting about during the coffee hour. Like, the insect recommendations are the easy part, right? Like, the hard part is like talking to the stakeholders and getting adoption. And that's sociology and psychology and economics. So making sure we're working with economists and getting the economic components. And then I really have always wanted to get the natural enemy counts incorporated into the economic thresholds. And I feel like we're really close to that. So this is just an example of the type of field trials and types of results we could get from that. And what I just want to point out is I'm going to talk a lot about these two different active ingredients, chlorantriniloprol and bifenthrin. Bifenthrin is our most commonly used pyrethroid, much more of a broad efficacy. Chlorantraniliprole is a diamide. It's a newer active ingredient, more of a targeted active ingredient for Lepidoptera. So, and the point of that was that we did see a difference. The control here had the highest number of natural enemies. Chlorantraniliprole was less than the control, but it was still a lot better than the bifenthrin. The pyrethroid bifenthrin was the worst in terms of reducing natural enemy abundance after spraying. This is now a project from Dr. Ana Lima when she did a sandwich PhD in my program. These are some toxicity bioassays that she has done with our pest, our Western bean cutworm Stryocosta albicosta, the young larvae here, and also the larvae of Hippodaemia convergens. So now that we've really identified that lady beetle as being a key player, we can incorporate that into our toxicology studies as well. She tested a lot of really interesting essential oils and different ethanolic extracts of different bioinsecticides here, but also had bifenthrin and chlorantraniliprole are two kind of conventional ones that I've really compared here. She also did some behavioral bioassays using the LC20 and applying that to the LC20 of the pest and applying that to the larvae of the lady beetles, and then tracking them with the EthoVision system to look at like walking speed and distance moved to look at behavioral sublethal effects. And really what I want to point out here from the toxicology studies is the differential selectivity index here, right? So this is the ratio of the LC50 to the pest compared to the ratio of the LC50 of the natural enemy. You want this to be more than 1, but this is actually showing bifenthrin is more toxic to the lady beetle than to the pest. Chlorantraniliprole is almost 59 times more toxic to the pest than the lady beetle. This is what we want. And then, holy cow, this is one we're trying to import from Brazil right now to do more studies on. This is a bioinsecticide that's commercialized in Brazil and had a really awesome result for us, and it's something we're wanting to look into more. And this is just those behavioral results here. What I just want to point out, this is walked distance, like the matrine, that's that awesome Brazilian one, did not reduce walking distance compared to the control. And chlorantraniliprole also did not reduce walking distance. However, the bifenthrin really did. And then similarly to speed, you can see that the walking speed of the larvae did not change between the control matrine and chlorantraniliprole, but it was pretty reduced for the bifenthrin. So this really kind of leads us back around to the little bit of that economic question again of like, all right, great. Like we're chlorantraniliprole. It's really expensive compared to bifenthrin, right? Like there's a reason why all the farmers are not just going to adopt it immediately. So we really have done some work to also think about how do we figure out the scenarios that make sense to spend more money and get an economic return, kind of an economic and an environmental return on investing in a more expensive product. And so there are scenarios that I walk my stakeholders through. If you are in a situation, there's some interesting things I didn't have time to go into with the pest and plant phenology. There's a very specific growth stage of corn that's highly preferred. And so if you have eggs being laid at that preferred growth stage, you will get more injury. If you're in that kind of scenario where you're primed for a lot of injury, if you are observing natural enemies in your field, if you're not all the way to the end of the moth flight yet, and you might still have more moths coming in and laying eggs after you need to spray an insecticide, using a less toxic insecticide that's more expensive but will maintain more natural enemies in your field, can allow those to kind of clean up those later laid egg masses that come in because there's still moths flying. So these are really the scenarios that I walk folks through to try to get that impact. And then that's sort of the next step, right? Taking all of that and communicating it with the stakeholders and trying to have that impact, trying to change knowledge, try to change behavior. I also wanted to share a little bit. Okay, I'm having trouble doing math about what time it is. Okay, I have like 1 minute. I wanted to share a little bit about this. This is like taking extension to like maybe a little bit of the next level. This is a peer-to-peer learning competition. It is called Testing Ag Performance Solutions. It was originated at my Extension and Research Center in North Platte, Nebraska. It is a farm competition. So teams make real-time decisions within the season on replicated plots at our research farm. So they will choose what hybrid they want to plant. They can choose, it's a very focused on nitrogen and water use as well. So they can choose like how much nitrogen and when and in what methods they apply the nitrogen. They can choose how much water they're going to irrigate each week. And they also get real-time data to plug in to make that decision. So there's like water moisture sensors and imaging and crop scouting and different things that are provided to them to help them make those decisions. And then the decisions are actually implemented on real replicated plots in the field. And at the end of the year, they get prizes. It's very competitive. They're very into it. There is a reward. There is a prize for highest yield, but the financial award gets scaled down for their efficiency. So if they just put like 300 pounds of nitrogen and 25 inches of water, you're not going to get anything. Like you use too many inputs, it doesn't matter what your yield was, because the most important thing is input use efficiency. That's what they really get prizes for. And they also get prizes for profitability, because we put the economics of all these decisions in there, and then they actually market their grain at the end of this season too. And we've added an insect decision to this competition. So we scout their fields, we send them scouting reports, we send them, here's all the information you need to know about economic threshold. Here's all our information about our insecticide efficacy trials. And there's costs, they could choose these different products, choose your rate, here's how much it'll cost you, and then we apply that as they've decided. And this is, you don't really need to understand this graph, just that everything overlaps with 0 here. Like this was last year, we were not above economic threshold, and yet some people still sprayed and they all lost money. I was a little sad that people still sprayed, but it was a very good illustration of like this economic threshold worked. You lost money. It also was a very stark indicator of we have some more work to do with our education about why are we using thresholds and that sort of thing. Yeah, so this is just sort of like coming back around to this. Where have we moved the needle a little bit about understanding? We've improved trapping and scouting. That's another thing I didn't have time to think about. We've decided really to focus on the generalist predators. We need to maybe work with a different trichogramma species that's better adapted to our arid climate. We didn't really get good efficacy of our trichogramma. We are still kind of looking at this pyrethroid resistance that's happening and how do we apply it better. And then some other cool things of like, could we apply some entomopathogens to the soil and maybe have some mortality to overwintering? Yeah. And I think like, I'll just say my favorite quote from like a survey I did for Extinction Impact is, lady beetles are good to have in the fields. And so I feel good that the farmer took that away. So I just want to say thank you. Of course, this has been the work of 87 students, technicians, postdocs, undergraduates, interns, sandwich students, amazing almost 12 years of folks coming through my team, and great partners. And yeah, I will be leaving Nebraska next year. After this amazing 12 years, I am really excited, as you all, if you remember from the beginning, I'm from Kentucky, and I got my PhD there. And I'm really excited. I got 2 little kids and really happy to be able to move home and work with some amazing colleagues at University of Kentucky. And a little excited and scared to switch commodities, to leave corn and to be working in fruit and vegetable production. So applying very similar approaches, I think. It will be an extension and research position, applied ecology and IPM, very similar to what I've been doing, but different insects and different crops, and I have a lot to learn. I have a lot to learn from, you all have such amazing strength in these areas here at Florida too, a lot to learn from you guys. And around ESA time, I think I'll have an ad out that I am looking for a master's student and a technician who would start in July or August at Kentucky with me. I don't start that position until July 1st. So a little bit of time to wrap up at Nebraska and move across the country. Okay, thank you so much.